Hello guys, you're welcome to today's lesson, and today we're going to be talking about ubiquitin proteosome system. Now, proteins are being synthesized in the system, and sometimes newly synthesized proteins may carry some errors and so are defective, maybe due to translational errors. The proteins might be defective. Now, how can the cell recognize these defective proteins and destroy them? Because if they are not destroyed or removed from the body, there is a problem. So, how does the cell recognize defective proteins and destroy them? This can be achieved using the ubiquitin proteosome system. And in the ubiquitin proteasome system, there are three enzymes involved. There are three enzymes, and the three enzymes are E1, which is called ubiquitin activating enzyme. That is E1, ubiquitin activating enzyme. Then E2, ubiquitin conjugating enzyme. That's E2, ubiquitin conjugating enzyme. And E3, ubiquitin ligase enzyme. So these are the three enzymes involved in ubiquitination. The three enzymes involved in ubiquitination. Now, before we look at the role of each of these enzymes, one thing we should know is ubiquitin is a molecule that once it is attached to a particular protein, that protein is meant for destruction. Like ubiquitin is a molecule that sends signal for destruction. So any protein that is attached with ubiquitin, forget it, that protein will be destroyed. So first, once the cell recognizes that there is this protein is defective, it will look for a way to attach ubiquitin to it so that the protein will be destroyed. Now, how is ubiquitination, which is attachment of ubiquitin, done using these three enzymes? So let's look at the mechanism. All right, so let's look at the mechanisms of ubiquitination. Now, this molecule here, this is ubiquitin. This is my ubiquitin. Now, this is not the full structure of ubiquitin. We just use UB to represent ubiquitin, and this is the carboxylate side of ubiquitin. This is the C terminal, the carboxylic acid side part of ubiquitin, and this is where we're interested in. So the first step in ubiquitination is activation of ubiquitin. So we activate ubiquitin with ATP, adenosine triphosphate. Now we take one phosphate group, we take adenosine monophosphate from ATP and attach to this, which will give AMP is attached to ubiquitin now. So now let's look at it. If you remove AMP from ATP, what is left? Pyrophosphate, right? So pyrophosphate, 2-phosphate is giving off. So this is our activated ubiquitin. Now the next thing is E1 comes in. E1, remember our E1 is what? Ubiquitin activating enzyme, right? It comes in and removes AMP, displaces AMP. That is what you're having here. So we just remove AMP and add our E1, and it is linked by a sulfide bond. Now that is the work of E1, but E1 is still here. The next thing is E2 comes in and removes E1. E2 is ubiquitin conjugating enzyme, right? It comes in and it removes E1. So E2 is attached in place of E1. We are now having E2. The next thing is E3 comes in. But what E3 is doing, E3 carries the whole of this complex now to the target protein. So this is still this molecule. E2 is still attached. Then the target protein, the protein that is meant for destruction, the defective protein is here. So E3, which is ubiquitin ligase enzyme, carries this complex here to the defective protein. And after that, after carrying it to the defective protein, the next thing is E2 will go out, E3 will go out, and the ubiquitin now will now attach to the defective protein. So we're having this is ubiquitin, and this is the target protein, the defective protein. Now any protein that is attached with ubiquitin is meant for destruction. Now you must note that there might be polyubiquitination. Yes, it's possible. That is, more than one ubiquitin can attach to a particular protein, and the more ubiquitin attached, the more the signal for destruction. That is, if I add 2 ubiquitin, now the signal for destruction will be more than when I add just 1 ubiquitin. Do you understand? So basically, these are the roles of the 3 enzymes involved. Ubiquitin activating enzyme, we activate the enzyme, we activate the ubiquitin, and after that, after the activation, E2 comes in, displays E1. E3 now carries it to the defective protein. This is just the complex where defective protein is found. Then E2 and E3 goes out, and we have our ubiquitination done. Ubiquitin is now attached to the ubiquitin, the target protein. Now, after attaching to the target protein, what next? What then happens after the defective, the ubiquitin have attached to the target protein? How is the protein destroyed? Alright, so how is the protein destroyed? The protein is destroyed in the proteosome system. The proteosome system. And the proteosome system, where the defective protein is destroyed, is made up of 2 apparatus. We have the 19S proteosome, or the regulatory subunit, and we have the 20S proteosome, which is the catalytic subunit. Now we have 19S proteosome subunits here, 19S subunits here, that acts as a regulator, regulates the protein that comes in to ensure that it is only defective protein, it's only ubiquitinated protein that comes into this catalytic subunit. Any protein that gets into this catalytic subunit will be broken down. It is in this catalytic subunit that the defective proteins are broken down. Now, when 19S proteasome combines with the 20S proteasome, we have the 26S proteasome system. So the proteins are degraded, are destroyed in the 20S proteasome, and the proteins are broken down into shorter chain amino acids. What are those amino acids used for? Those resulting amino acids, what are they used for? Food for thought. Let me know in the comment section the answer to that question. If you have any question also, please feel free to drop it in the comment section. And please, as you watch, help me subscribe to this channel. By subscribing, you are encouraging me to drop more videos, which will also help you. Thank you very much for watching.